Really happy to be here. I'm going to go through my talk fairly quickly because there's two of us. I'm talking about microbes and bees, and then Dean, my husband, is going to talk to you about queens and breeding. So we have a lot of information to cover, so I'm going to go fairly quickly. If you have questions, hopefully there'll be time at the end for a little question and answer. If not, how many people are going to be able to come tomorrow for the workshop? Great. So if there isn't time tonight, you can always find me tomorrow when we can talk more. But microbes and bees, I got interested in this topic almost 6 years ago, and I'm not going to take time to go into the details of how that all happened, but once I realized there was a connection between the microbes and the bees and that the microbes were fundamental to the bees, I got very excited and have been reading as much as I can to learn more. So this is a basic introduction to the topic. We're talking tonight about primarily yeast, bacteria, and fungi that are part of the bees' lives, both in their bodies and in their food supply. Viruses I've put down here because they are part of the whole system, but I'm not going to go into too much detail about those tonight because they're complicated and hard to understand. So mostly we'll talk about the yeast, bacteria, and fungi, which are the invisible members of the bee colony. We can't see them, but they perform fundamental roles in the hive. I like to start by thinking about the planet, because if we were aliens and we just happened to see this ball in the sky and it looked like that, we really might not have any idea of what was going on under the surface. But because we're humans and we understand the planet at least to some extent, we can see here in North America, South America, we have some ideas of how cultures live on the planet, how ecosystems work, how animals live, people, populations of people, ideas that populations might be clustered more around like waterways or coasts. There's a lot of information looking at the planet that we all have just from being inhabitants of the planet, even though from this perspective we can't really see what's going on. Get down a little bit closer, we have like an aerial shot of West Palm here. Looking at this, if we were aliens, we would have no idea what was going on. But because most of you all live around here, you probably have some ideas about what it means to see the water, what it means to see the streets. You know, you can't really see that those are houses, but you probably have some idea of dwellings and what goes on in the dwellings, and you can imagine populations of people moving through the streets, etc. So I like to keep these kind of pictures in my mind when I'm thinking about the microbes, because we can't see them activated in the hive, we can't see them doing the work that they're doing. But if we imagine that they're performing tasks and functions and all sorts of things in similar ways to the way all the other life forms on the planet are performing, we can kind of use our imaginations to sort of animate them and make them more real. So while I'm talking, you can kind of have a picture in your mind, like inside the bee body and the bee bread, there's all this life going on that's parallel to all the other life that's going on in the planet. It's just in very tiny, almost invisible colonies. So all living things need a suitable environment, right? You need a place where you can actually have shelter that's going to support your biology. You need some kind of sustenance, food supply, survival skills, and these three things you need not only on an individual level, but on a population level, and some kind of means of reproduction. Otherwise, the species is going to die out. And microbes are the necessary foundation of larger life forms. Most of the life forms on the planet are microbes. They're everywhere, from the depths of the ocean to hot thermal places underground, boiling hot, acidic, all kinds of pHs. Almost anywhere on the planet that you can think of, there's some kind of micro life that lives there. Again, they make up the bulk of life on the planet. They occupy and adapt to advantageous niches. So just like all other life forms, they're looking for a place that they can hang out and live and prosper. And they're very good at extremes. They have co-evolved all life on the planet, and they're nature's ultimate recyclers. So almost anything on the planet that's available for food, some kind of microbe will have figured out how to make use of it. And they make the continuation of life possible. So without the microbes, I think if all the microbes on the planet ceased to exist within a couple of days, all life would stop, because the microbes are at the bottom performing all these minute tasks that basically make all the life above them possible. They prepared the planet for all other life. populated the planet. They were our first residents, support all the larger life forms, and live on in our cells as mitochondrial DNA. Like all of us in our cells have parts of what used to be bacteria that now are parts of our cells. They compete without mercy. So again, you have populations that are trying to survive. There's competition for resources on every level. They're utilizing unutilized food, unutilized waste. They exist in complex adaptive cultures and systems. And this part is really important because as sort of all life forms have to adapt, right? You have resources, and as resources get used up, waste is coming out, something else is making use of that. But as demands are being put on a population, they have to adapt to it or cease to exist. So there's a sort of stretchiness to the whole system where as things become available, other things move in to use up what's available. As things get depleted, things have to move around and find other sources of goods. There's constant movement to the system and sharing of resources back and forth. Microbes can create colonies to act as larger life forms. So as individuals, they don't really work. You can't really have much going on with one bacterium. But as they multiply and create colonies, now they have almost the force of a being to get the jobs done. And they're always balancing. So we have in this little drawing, the spiral can represent an organism or it can represent a group or a population. There's eating, consumption through eating, or whatever we call eating, taking things in, metabolizing, whatever that thing is that's taken in for the life form to go on. And then there's kind of a spinning off of something that would either be toxic stuff to one thing or useful stuff to another thing. And then taking in again. So this is constantly going on and requires competitive cooperation. Because if one being or one group takes up everything, at some point they're going to run out and that's the end for them. So there has to be some kind of give and take through all of the populations to share the resources and be able to give back. Otherwise the system can become static. And this is a bigger sort of illustration of the spinning off of consumption, metabolizing, giving off waste that becomes then useful to something else or toxic to something else. Locally and globally, the microbes are the unseen, unappreciated masses. I sort of think of them as the janitors of the world, doing very, very important tasks. You can imagine what would happen if all the janitors suddenly didn't exist, how much waste would pile up on the planet. They're performing vital tasks that affect the bottom line. So even though the tasks that they're doing are, we can't really see them, they're supporting everything that's happening above them. They're outsourced by larger organisms for cost savings and adaptability to market forces. A lot of times these microbes have evolved along with the larger organisms and do tasks that save energy for the bigger organism. And they kind of coexist together and help each other. The bigger organism giving resources and the microbe performing tasks. And they fill in gaps in the system. Nature abhors a vacuum. There's always some place that the microbe can occupy and be useful. And mostly they're beneficial or innocuous when the environment's stable. For all the microbes that live in the world, all the bacteria, all the yeasts, all the molds, very few of them actually cause a lot of problems. Most of them are actually coexisting with all the other life forms and helping them out. So in the honeybee colony, we have a simple system with very complex needs. Pollen, nectar, water, and plant resins are basically the inputs to the hive. But the needs that they're filling are nest structure, brood rearing, preserving food, physical defense of the hive, and microbial defense. And the microbes are basically at the bottom performing tasks that allow all these needs to be met. The bee-related microbes have co-evolved with the bees and plants over millions of years. Some of them don't exist outside of the bees' bodies. They've co-evolved to live only with the bee, and if they were removed from the bee, they couldn't survive. They're an integral part of the honeybee anatomy and biology, must be included as vital members of the colony, rarely cause disease in the colony or in the bee. They're parts of the bees. plant-associated microbes. These, what do the bees take in? Pollen and nectar from plants. That's their whole diet. So we can't think about microbes without also including the plants. Plant-associated microbes, parts of the bees, because of that. They recycle nourishment through decomposition. They feed root systems. They can be defended against by toxins produced by plants. Plants, the propolis is basically the plants, or the resins are the plants' way of defending against microbes. And the bees appropriate those toxins for their own defense. And that defense can be physical or microbial. So this drawing illustrates sort of this web of sharing where bees have the microbes that are associated with them. Water, there's microbes in the water. The bees take in the water. The plants take in the water. We have microbes that live in the roots of plants. And in some plants, a huge amount of their energy goes out as sugars through the roots to feed the microbes that are then performing tasks at the root base to break down and feed nutrients back to the roots. So that's a huge part of what's going on. And in turn, the pollen has a lot of yeasts and molds that contribute to the hive. And the nectar also has some levels of microbes in it as well. So there's this constant sharing that goes in a big loop of the web. And we're all bee-centric here. So bees are the thing that we think about, but the bees, again, they're just a tiny part of the giant ecosystem of the planet. So the three most important things that we'll talk about tonight are how the microbes help the bees defend against disease, help the production of their food supply, and help them digest and assimilate their nutrients. So in the colony, we have our cast of characters: queen, worker, and drone. The microbes, we talk about them as the invisible cast. So that's an important thing to keep in mind, that they're definitely part of the whole picture. So one of the most important, everybody here been hearing about human gut microbes and how important they are. People have a general idea of that. Every organism with an inside has a gut. So we can be thinking, sort of paralleling what we know about human gut microbes to the bees. The gut is the pathway where nutrition enters and waste exits. You have to have some way for things to come in and things to go out. And through the gut, basically the gut is feeding the rest of the body. So the nutrition's coming in, and everything's happening in the gut to then feed all the other parts of the body. The gut microbes control fundamental aspects of the organism function and vital to the health of the organism. And the microbes are critical to the gut function. So I don't know if anyone here has heard of the problems with the C. difficile, the bacteria that can overtake your gut. If you have taken intravenous antibiotics, you can wipe out all the good microbes and you're left with a virulent microbe that can basically kill you. That's a good thing to think about as we're talking about this, because all those microbes that got killed off, they're performing essential tasks in the body for pretty much everything. Now they're linking it to depression, obesity, allergies, almost anything you can think of, you can come back to the gut. So here we have our honeybee anatomy. The hypopharyngeal gland and the mandibular gland, those are used in the production of food for the young larvae. The honey stomach is the place where the bees carry the nectar back to the hive and then regurgitate it back out to either feed the young or to store for honey. The little blue thing between the yellow and the orange is their proventriculus. It's a little valve that separates their crop from their digestive system. Then there's the midgut. The ileum, the green thing, is where a huge amount of important microbial activity takes place for the absorption of nutrients. And then the rectum is where the wastes are stored for elimination. And it's really important that the bees be healthy enough to have really good glandular and muscular development. So you can see the wing muscles there. For them to perform the tasks in the hive that they need to feed the young and get the food supply in and all the sort of work that they need to do to sustain the colony, that requires really strong flight muscles, also for heating and cooling the hive. Vibrating the muscles in the winter for warmth, and being able to fan for flying and for cooling in the warm weather. And glandular development, very important for feeding the young larvae. That's where all of the brood food is being manufactured in those glands in the head. Now, the microbes, there's microbes that are associated with all of the parts of the gut. One of the most important parts, again, is that green section, the ileum. And we've been following the work of a lab that was at Yale for quite a while, quite a few years, I think maybe 5 years or so, and now the lab has moved to the Austin, Texas campus of the University of Texas. Nancy Moran has that lab, and they're doing a lot of work on fundamental understanding of gut microbes in honeybees. And they're finding that in the ileum, in that green area, here's a little cross-section of one of their slides. This is a cross-section, so there's like 6 major folds that are very extreme, and where you see the kind of turquoise blue, the greenish blue, those microbes have co-evolved with the bees and don't live, they're bacteria that don't live anywhere else on the planet. They live in the gut of the bee, and they're performing vital functions to get nutrients from the bee's food into their bodies. And those microbes live in colonies where they're basically like attached to the cell walls of the gut. And if you try to remove them and grow them outside of the gut, they can't live. Like, it's almost like the microbes and the bee gut are one in that place in the bee's body. It's like a whole little world in there. This slide I took from my family values talk, but one of the things we've been thinking a lot about is the human fecal transplant. Has anyone here heard about that? I talked about the C. difficile a little earlier. If the gut microbes in your body are wiped out, one of the most effective ways they've found to bring people back to health is to take a poop sample from, they say, a friend or a loved one, someone that you're close to, probably to remove the yuck factor. But basically a healthy stool sample. They mix it with distilled water. They run a tube down your nose, past your stomach so the acids in your stomach don't kill the microbes, and into your gut. And with that method of basically re-inoculating your gut, they have a 95% success rate of bringing people back to health.
And those are people that...
People that basically have tried everything and they're basically on their deathbed. So to go from being on your deathbed to like a 95% success rate to be brought back to health from just a fresh poop sample is kind of an amazing thing. So before I even knew about the fecal transplant, I think it was the fall of 2008, I was first talking about the microbes, and I started to think about the practice we have of giving a weak hive a frame of brood and young bees. Has anyone here done that? You have a weak hive and you say, oh, well, give it a frame of bees from a really good strong hive. And we always think we're boosting the population. But I started to wonder, I wonder if what we're really doing is boosting the microbial population. Maybe the guts of those bees that are from the really strong, healthy hive, maybe those gut microbes are better, and they're able to share now with the bees in the sicklier hive and kind of re-inoculate the hive. And in talking to Nancy Moran from the lab that's now in Texas, one of the things she's told us is there's way more going on in the back end of the bee than the front end of the bee. Like basically in that anatomy drawing I showed you, the honey stomach has some lactobacillus in there. The midgut, not much going on in there because it's constantly shedding. But once you get to the ileum and the rectum, that's where all the activity is. So how is it that the bees are getting those microbes? Like, where are they coming from? When they look at the bees after emergence, not much going on. Basically the bees are born sterile, and then they get inoculated over a 3-day period. And the microbes they start out with on the first day aren't the microbes they end up with on the 3rd day. And none of the microbes they end up with are the microbes that they're getting through either chewing through the wax when they emerge or being fed by the other bees. So somehow they're getting inoculated. How is it happening? So I speculate that those young bees, when they're first born, they're the house bees, right? Their first job is house cleaning, licking, licking, licking everything. Possibly there's a lot of poop consumption going on in those first few days of their life so that they can get their guts inoculated. So we have this little censored slide here, but I honestly think that's what's happening because there's not really another way to explain how the microbes are ending up where they end up. So, the honeybee nutrition cycle. We can call it a nutrition cycle or a life cycle or whatever, but we start here with foragers. Foragers bringing the food, the pollen and nectar back to the hive. They pack the pollen into the cells to make the bee bread. The young nurse bees between 4 and 14 days old, they consume the bee bread to make bee milk. They feed the young larvae. Those young larvae are emerged, and then they go on within 4 days to start feeding the next round of brood. So you have a very tight cycle there. And if the bees aren't getting the nutrition that they need, you could have generations that aren't quite as healthy that are now responsible for feeding the next generation of young larvae. So it's really important that, especially the pollen that they're gathering, first that they have real pollen, and then that the pollen's in good shape. I'm going to talk about that in a minute. So unlike humans where, you know, it might take 15 to 20 years for a woman to bear a child, the bees are doing these cycles within matters of like weeks. So you have a very tight system there of being strong enough and having the right nutrients in your system that you can then pass on to the next generation. Bee bread is one of the most important ways. So we have the gut microbes. Oh, going back to the gut microbes. So not only are those microbes helping the bees to assimilate and digest their food, but they also have a way help the bees to defend against disease. So there's, in the lactobacillus in their saliva, they can basically, I'm going to go back a minute. When the foragers are gathering the pollen, the first thing they do is they wet it with saliva from their mouths. And that saliva has some of that lactic acid bacteria from their honey stomach, and it inoculates the pollen. There's yeast and fungi on the pollen that activate with that bacteria and start to ferment the bee bread. So when they're consuming, even within an hour of their gathering that pollen, the bees aren't consuming pure pollen, like fresh pollen, they're consuming fermented pollen, because that fermentation process starts right away. And that is what makes bee bread different from pollen, is that bee bread is actually a fermented product. So you know, the bee bread, if you see fresh pollen, it's like light and dusty, or kind of like,
Nuggets?
yeah, or just the texture of it, it's not shiny. It's kind of powdery. Once they make it into the bee bread, the way you see in the cells there, it's undergone its fermentation process and it's capped over with a little bit of honey. That's how they store and preserve it. So this fermentation process is really important to the final product. The pollen and the resident microbes are collected and inoculated. There's a fermentation process that begins that's extremely active, and that happens within the first few hours of the bees gathering the pollen. They store it. There's intense activity for 12 hours, and then basically there's a microbial succession where each microbe that's predominant is kind of setting the stage for the next round of microbes. And after, so you start out with your like lactic acid, and then you move into your lactobacillus, and then you end up with the bee bread, which can be stable for the bees for the winter. And it takes about 2 weeks to end up as that final bee bread, but meanwhile, as they're consuming, even after that first hour, the fermentation has started. So we like to call bee bread a pollen pickle because it really is more like a fermented pickle. It's not a vinegar pickle, but it's something where the pickling is happening through the fermentation. And that fermentation produces, basically there's vitamins and increased protein. Not only does the fermentation help to break the outside of the pollen to allow the nutrients out, but it actually changes the chemical compound of the pollen. There's antibiotics that are produced, vitamins, enzymes, fatty acids, sterols, and some of these are unique things that the bees can't get anywhere else in their diet. So when you hear that bees, they can't survive for more than maybe 2 rounds of brood rearing without real pollen, that's because there's things in the real pollen that they just can't get anywhere else in their diet, and those are facilitated by the microbes. So I'm going to go really quickly through the honey. There's the unique lactic acid bacteria in the honey stomach that can also vary with nectar source. Bacteria in the nectar can protect it from mold and yeast fermentation. We all know honey has antimicrobial qualities, and honey can also detoxify natural substances in plants. Dilute honey can support microbial growth, and it's part of the bee bread process. So the honey, maybe not quite as much microbial activity is going on with the bee bread, but still really important to the bees. So the honeybee social microbial culture is using simple inputs, remember our pollen, nectar, water, and propolis, to provide for complex needs. It's heritable, persistent, and adaptable. The microbes, changes that they undergo in the hive due to antibiotics or other things that can alter the microbes so that their offspring have those same resistances or those same qualities. And it transmits vertically from generation to generation. So when we're thinking about microbes, like with humans, we're starting to learn that maybe our microbial cultures, especially in our guts, might be more important to our health than our genetics. This might also be true for the bees. Little quote that I like. It's helpful to remember that in nature, friend and foe, cause and effect are not always distinguishable. It's interesting to think that it wasn't maybe just a little more than 100 years ago when Louis Pasteur discovered bacteria, and bacteria was seen as the enemy. And now we know that things are way more complex than that, and that at least for humans, bacteria contribute a lot to our well-being. So microbial imbalance expresses in disease. We know that foul brood, the two foul broods, American foul brood and European foul brood, are both bacteria. Chalk brood and stone brood are both funguses, and Nosema is a microsporidian, which is kind of not unclear what Nosema really is, but it keeps getting reclassified. Viruses, I have a lot of question marks there because the viruses are so strange and ever evolving, so they're there because we need to think about them, but I'm not sure exactly how. There's a lot of discussion right now about viruses, especially as they tend to be vectored by the mites. So this drawing, this is sort of a way to illustrate pressures. We have the yellow, the green, and the red. We have our yeast, our molds, and our bacteria. Those are competitive within each other. So you can have bacteria that are competitive with each other. Molds that are competitive with each other, yeast that are competitive with each other. Everybody's sort of back to our original thing of competing for resources. All these populations are trying to get the goodies and crowd each other out and get what they can. There's a lot of competition. Then we have pressures, like say we decide we're nervous about a bacterial infection in the hive and we decide to give it, say, terramycin. Well, the terramycin is going to put pressure on the two foul broods. But it's also going to put pressure on the other bacteria in the hive that we might need for other roles in the bees' bodies and in the colony. We know from laboratory studies, and a lot of this information comes from lab work, that sucrose feeding or high fructose corn syrup feeding can put pressure on both molds and bacteria in the hive. And once there's pressure on those, that allows yeast to take over or become more strong. Fumagillin that's used to put pressure on Nosema, the same, the fumagillin's basically produced by toxins that the stone brood fungus puts out. You know, penicillin is a mold. So there's different things that are putting pressures on the hive without being manufactured, and some of these things we can manufacture out of those toxins. Fungicides are probably the biggest one that I'm thinking about now, because when we talk about pesticides, pesticides tend to get lumped into one category. But fungicides are specific to fungi, and they're allowed to be sprayed on open blooms. Whereas most pesticides, there's label requirements for when you can spray them. Fungicides, they don't affect the adult bee. They don't kill an adult bee. But they might be having a very big impact on the pollen that those adult bees are collecting and needing to process into food for the young larvae. So if those molds and those yeasts are being shut down or impacted by the fungicides, you can end up with a food that looks like it's supposed to look, and the bees are treating it like it's food, but it might not have the essential nutrients that the bees need to get that young larvae to grow and develop properly. Organic acids, we know, are very detrimental to molds and yeasts. Now, we know in the lab that they can show that they affect greatly how molds and yeasts develop and grow, but how does that translate into the field? A lot of this stuff It's very hard to do like field studies to figure out what cause is related to what effect, but we know that in the lab, these things all have an effect. Yeah. Okay. 10 minutes? Okay. So again, we'll talk a little bit, followed on the war on microbes. So if we're thinking about our hives and we're thinking about treating or not treating or what to do, first, the fungicides may be more harmful than the pesticides. And that's just something to have in mind when you have your bees in your agriculture. Because in our minds we think, oh, farms are such a great place to keep bees, but they might not be the best if things that are being used are affecting the pollen. Antibiotics can change the climate of the gut microbes and disrupt the workforce in the gut. Targeted killings, we want to go after one thing, but they can result in collateral damage that we didn't intend. And the field is more complex than the war room. We have a lot of things that we can see in a laboratory, but how are they translating into the field? And these, I got an email from a guy in Philly saying, I really hope when you come to talk on Sunday that you give us some practical information, because like hearing how great the microbes are, that's great, but like, I want real information. And a lot of this stuff, there is no real information. It's just, we can speculate, but we don't know. Like if we put formic acid in the hive, what effect is it having on the bee bread? We know if we look at it in a laboratory, it's having an effect on all of those yeasts and fungi that support the bee bread. But what's it actually doing in the hive? And how is that impacting the health of the bees? Like these are studies that really haven't happened yet that are looking at like practical applications. So I'm just giving you a lot of information to think about and hope that you all get excited and want to learn more. And maybe at some point there'll be ways to figure out what exactly the metrics are between treating or exposing bees to certain things and what the final outcome is. And every decision has a cost. So no matter what you do, there are going to be positives and negatives. So the take-home message here is an increasing awareness of the importance of microbes for healthy, well-nourished bees, and that healthy, well-nourished bees are the best defense against disease. And if your club has a library, this is actually a really interesting book. This came out a couple years ago. It's edited by Diana Cimitaro from the Tucson Bee Lab and Jay Yoder from Wittenberg University in Ohio. And a lot of sort of nerdy scientific studies, but rewritten in a way that are easier for an average person to understand. So if your club, this is like an $85 book, but it's a really good book to have as a reference, and you could all just, your club could get it and you could all share. Now, Martha Gilliam, she's a person to really know about. She's kind of the first person back in the 70s that started really looking at the microbes in the bees. And we had an opportunity to go to Tucson and basically scan all of her papers. And they're on our website in a archive. She was using culture-based techniques, though, anything that could grow in a petri dish. And at that time, in basically the 70s through the 90s, she identified over 8,000 microbes. Now with DNA sequencing, the researchers are able to look more closely at the microbes and try to figure out which ones are actually the big players and which ones are kind of peripheral. And that's a lot of what's going on at the Moran lab now. So this is the interesting part for me, the unanswered questions. How exactly do the bees become inoculated? We have our suspicions about the poop and the poop eating, but is that really what's going on? The Moran lab is actually looking at the difference in the microbial cultures of treated versus untreated bees, and they're finding a lot of interesting things, especially around the antibiotics. What about queens and drones? Are their microbial cultures different from the workers? What roles might their microbes play? Are they the same microbes that the workers get? Are they different? Nobody really looks at the queens and the drones. So this is another big question. Do microbial cultures affect the fertility of queens and drones? There's been a big talk for the past few years about fall-off in fertility of queens. A few years ago, Dave Tarpey came to our bee club and he had a chart, actually the chart's in this book, showing percentage of nosema in queens over like the last 50 or 60 years. And all of a sudden it got to like 2010 and there was zero nosema. And we asked, like, well, how could all of a sudden you've had 15%, 30%, 20%, how come 0%? And he said, basically the breeders are feeding the snot out of them with fumagillin. And you have to wonder, like, is there something that that's doing that's affecting the fertility? I don't know. What role does the feeding of drones by the workers play in the microbial cultures of hives? How are the drones getting inoculated if they're being fed by the workers? Do drifting drones impact the microbial cultures of hives they visit? Is that a way that we can move microbes around? Drones are just hanging out in the hive. Strange drones come in. Are they pooping? Are the young workers cleaning? Like, is that another way for microbes to travel? And a big one for me is, what's the relationship between the fungi that support the root systems of the plants and the resulting quality of the nectar and pollen? We have a friend who's basically a plant expert in Georgia, and I asked him, are there any studies that even are looking at this and show this? And he couldn't find any for me. So that's another field that I think would be really interesting. Like, is there a difference between the food that the bees are getting from plants based on what the microbes, how rich the microbial cultures are that are supporting them? So here's a few resources. We have at our website, beeuntothers.com, a bunch of microbial reference articles in the Gilliam archives. A little bit more information about the Yale project on the bee microbiome project. Nancy's since moved on to Texas, but I believe that website still has the information about that lab. The book I just showed you, and anybody that's interested in the microbes in humans, if you Google the Human Microbiome Project, hundreds of articles that are incredibly interesting. No time to really get into it today, but just I'll tell you a quick one. Like, when we're born, we're sterile. We get inoculated when we're born just by the way we're born. If we're born vaginally, we get a whole different set of microbes than if we're born with a cesarean. And those microbes kind of set our guts for like the rest of our lives. Breast milk, you get a whole different set of microbes if you're breastfed than if you're bottle fed. And there's so many interesting things that just in the human body that start with like our inoculation when we're really little. So I encourage you all, if you're interested, to just Google Human Microbiome Project and you'll be reading for days. That's it.
Department, if you follow the best management practices here in Florida, they're going to tell you that you probably shouldn't be raising your own queens. Fortunately, I don't live in Florida and I don't have bees in Florida, and the ag department in Florida doesn't really have anything on me. So I'm free to come down from Massachusetts and tell you, you should be raising your own queens and try to help you learn how to do that. So I see that as an opportunity and a responsibility, and it's something that I take seriously. So I'm going to talk about queen rearing. I'm going to talk kind of fast, probably. This presentation and some other notes that I have, you can download from these locations. There's PDFs. So you don't have to I have some slides that are very text heavy. So I don't want you to spend all your time trying to copy stuff off the thing. I know it's a little bit, you're supposed to have very few words on here, but I wanted to present something to you that you could go back and read later and it would make some sense, not just be some signposts. Okay? So that second document, the Queen Notes, that's about a 20-page thing where I made up some quotes about queens from, Keith Richards and L. Ron Hubbard, and try to talk about queens from some different perspectives. And I'll just, it starts with, what is a queen? And there are a number of ways to think about a queen bee. The queen can be the mother of the bees in the colony. She can also be the daughter of the colony. She's an unstunted worker. She's the ovaries of the superorganism, and she's also the testes of the superorganism. She's the sperm bank of the superorganism. She's a monarch or a sex slave. She's the co-father of another colony. So I have more detail here, but I don't want to spend my time reading it. If you're coming to the workshop tomorrow, you can download this tonight and chuckle yourself to sleep and ask me some questions about it in the morning if you like. And again, we'll be able to talk a lot about this stuff much less formally tomorrow. So why should you be raising your own queens? James Powers is really the first of the big migratory beekeepers who kind of invented almond pollination as a way of beekeeping, which now drives the industry. And he said that queen rearing is too important a task to leave to someone else. And I think that's true for every one of you here. I'm hoping that by the end of tonight, all of you are going to be thinking about raising your own queens this year, even if you only have one hive. It's an important and rewarding dimension of keeping bees. It allows the beekeeper to build on the genetics of the hive rather than replacing them, right? You have a hive that's not doing so well and you buy a new queen, but that queen's mated. So if there's some aspects of that genetics that are well adapted to your area or that you like, you're really tossing them right out the window and replacing them with this mated queen that you don't have any control over. And certainly isn't hasn't been influenced by the area where your bees are, and not adapted to the area where your bees are. When queens are being raised by the bees or by the beekeeper, the most important thing is the nutrition that they're getting. As far as making a good queen, nutrition is paramount, then mating, and then the genetics of the queen. So even if you don't think that you have the gold standard of stock, you can still raise very good queens. You can raise high quality queens with minimal expense and work, and I'm going to talk about how you can do it without even finding the queen in your hive. You can raise queens. The beekeeping industry needs more actual bee breeders, and I'll talk a little bit about that, what I mean by breeding rather than producing bees. There's plenty of room commercially for raising queens. If you have a few hives and you start raising some queens and you're raising good queens, you won't have any trouble finding people to buy them. And all of a sudden you're in business being a queen raiser and a queen breeder. And most importantly, that once you know how to raise queens, you won't be able to stop yourself. Ask Sam, how often have you been grafting this year? Yeah, I think he told me every 10 days he's been grafting. So what can you expect if you're raising queens? If you prepare properly, you will be able to raise more queens than you can use. So when you're raising queens, you should raise more than you can use, because you can always share them, and things don't always work out as well. You know, you say, oh, here's the queen that I want, and sometimes she isn't there when you need her. A reasonably well-mated queen with moderate genetics will perform well, and probably perform as well as a queen that you're going to buy from somebody, probably better. And if you maintain your stock from year to year and apply some selection pressure to your operation, you're not just raising queens randomly, but you're saying, oh, I like this hive. Oh, this hive is, this queen has been doing well for me for 2 years. I think I'll raise some queens. You're going to actually make progress, and eventually, if you keep up with it, you'll have your own strain of bees that has identifiable markings, identifiable behavior. You'll have bees that actually have some characteristics that you can point to that you have helped develop. So, how queens are raised by the beekeeper. Queens are raised by bees as part of a system. That system has to be in place, primed, nourished, and well provisioned. Only after there is a queen-shaped hole in that whole system will that system produce a queen. And that's your job, is to make that system well stocked and then leave that hole in there for the bees to fill in. So what's required for raising queens? At least one strong hive. That's it. Now, if you want to raise a lot of queens, you need more resources than that. But you can certainly raise it. If you have one hive and you want to requeen it, or you want to make a split, there's no reason that you can't use that one hive to raise a queen or some queens for yourself. You have to get the timing right for making up cell builders, for the queens to mate, to check and not disturb the new colony. And we'll talk a little bit more tomorrow about the details of the cell builders, but you want to make up your cell builders a day or two before grafting, because I've done it both ways, and I've raised queens successfully by making a cell builder just on the day I'm doing it. But boy, when you open up that cell builder and they're ready to draw those cells out, you can almost feel it when you take the top off. You put the cells in, and they glom right onto the bees. So making them ready for it is really important. Now, if you're grafting, I'm going to leave some of these details for tomorrow. So, methods of raising queens. This is the simplest one. It's called a walk-away split. And in its simplest form, you split the hive in half. You make sure that each half has eggs and young larvae, because those are the basic resources needed to raise a queen. You don't know where the queen is, doesn't really matter. And you walk away. And whichever half of that colony that you've split in half doesn't have a queen will start to raise queens. The problem with the walk-away split is that it leaves the queenless half without a laying queen for more than 3 weeks. So that means that you have a month and a half before you get young bees coming in there. And that's quite a long time, and it certainly works. I've certainly raised some great queens with walk-away splits. I know Sam has as well. But it takes a lot of resources to just, you do a walk-away split, and you end up with one queen in that split that you made. So you've got a lot of resources into raising that one queen. The other thing about a walk-away split is it's something that you can do if you're afraid your bees are going to swarm. You can do a split, and you can kind of weaken both halves up to the point where you're forestalling swarming, and it's something that you can do as part of preventing a swarm. when I'm doing a walk-away split, or if you're doing it and it isn't your first time and you're thinking about it, you have to realize that there's these resources that are in the colony that you need. You need comb, you need stores, you need pollen. There's a laying queen in one half of it. You need brood for, so that you have young bees coming into each half, over that period of time. You have foragers. Foragers are important because the foragers are always going back to the old location, to where the hive was in the beginning. And you have, I lost my place, and you have house bees. We have these bees that are able to take care of the brood and to actually produce queens from the brood. So you try to take some care to appropriately split these resources up. And one example is, if you have a pretty strong hive, and it's in this location, you could take that hive and you could move it to another location, and you could leave at that original location literally a frame of brood with adhering bees. Now that doesn't seem like a lot of resources to raise a queen, but you had a strong hive there just 5 minutes ago. All of those foragers, even if they're still in that hive, they're coming back there. And they've got a lot of food coming in. And they're going to raise a queen, and they're going to do a good job with it. Even though you didn't give them, it doesn't look like you gave them many resources, you put one comb in there. It doesn't really mean much, except that you have all those foragers coming in. Those foragers are bringing pollen and nectar, are bringing that urge to raise a queen. And again, the downside of the walk-away split is it takes about 6 weeks before any of the bees in the split will be replaced by emerging brood. And that's a long time. That can actually lead to the bees deciding to swarm, that something doesn't feel right. So it's a good idea if you can, if you're doing a walk-away split, after a couple of weeks, to give them another frame of capped brood. Give them capped brood that's going to be emerging, so you have a little influx of young bees in there during that 6-week period. Because that 6 weeks is a very long time not to have young bees coming into the system. You leave a big gap in the generations there. Okay. So here's a picture from a walk-away split, and you can see these two very nice queen cells that these bees made just because their queen they didn't have a queen anymore. It took the queen away. Now you can take things a step further, and you can harvest. How many people have ever opened up their hive and unexpectedly found queen cells in the hive? It's something that happens. Well, you can take advantage of that, and you can say, I've got queen cells. I'm already a few days ahead of the game, or a week ahead of the game. I can use this opportunity to make a split and try to keep the bees from swarming, or if they do swarm, at least I've got, you might take a couple of frames out that have queen cells on them, make a couple of small splits, and then you've hedged your bets. And if one of those queens doesn't make it back from her mating flight, the other one does, and you can recombine them. Now, there's an important lesson here. If you open up your colony, where do you often find queen cells? You find them hidden in corners underneath all the bees. Well, when did you find them? After you shook the bees off the frame. Now, if you do that and the queen cell's still open and it hasn't been capped yet, things are pretty good because the larvae are very, very sturdy in that state. But you open up the hive. Even if you've been inspecting that hive on a regular basis, you find that hidden queen cell, you're not really sure if it was there the last time you were in the hive or not. And there's this period of time, about 10 to 14 days after they start the queen cell, that the wings and the legs are being developed. And actually the queen's fragile at that point. The cell is capped, but if that queen gets jarred around a lot, I've seen it on occasion. I've seen the virgin queen with like a front leg like this, and she's like walking like this. They're damaged. Sometimes I one time got a mated queen from a package producer that didn't have wings. I don't know how she got mated. I didn't find out if she did get mated. I didn't use her. But if you're thinking of that you might want to be doing a split like this, that you're going to want to take this opportunity if you find a queen cell to make a split, then open up the hive and be gentle with it, and don't shake all the bees off. Brush the bees off and look for those queen cells. Because once you've shaken the bees off, now you're looking at this frame and you say, well, gee, I don't know how old this queen cell is. I don't know if I damaged the queen. Do I really want to commit resources from this hive that could otherwise be producing honey or raising a queen through another manner just to find out if I damaged this queen or not? So I just caution you, if you kind of think that's the direction you want to go in, and it's something that I do. If I open up hives and there's queen cells, I tend to take that opportunity to split. If you think you're going to be doing that, approach the hive gently and approach the bees gently, and don't be too rough when you're shaking the bees off. Does that make sense? Okay. Now, we can take it a step further, and you can actually force the bees to make queen cells. And remember that the best queen cells, the best queens are produced out of abundance, not out of desperation. So what you'd really like is a hive that's so strong that they want to do what? They want to swarm. So, and if you force the cells, and I'll explain this in a second, you can time it, because you're actually saying, okay, now starting today I'm going to be forcing these cells. Well, now you know how many days old those queen cells are. And you can handle them appropriately and not handle them when they're fragile. These queen cells can be made available at the convenience of the beekeeper, not just at the opportunity that the beekeeper happens to open up the hive and there are some queen cells there. And the comb can be trimmed for better results, and I'll show you that in just one second. And this is a great way to raise queens. If you have never raised queens before and you want to raise a queen, or raise a few queens, you literally can crowd the bees. So I'll show you what I did here. And I gave a bunch of talks where I actually explained this, and I sat here waving my arms around and explained what you're supposed to do. And then I was going to give a talk. I think this was I think it was last August. I was giving a talk in Ocala. And I said to myself, well, instead of waving my arms around and explaining what it's supposed to look like, I ought to do it and take some pictures of it so I can actually point to it. So this picture's a little bit confusing because it has a wire right where I cut it, which wasn't ideal for the photograph, but so be it. And the idea is that, notice that this comb is kind of on the old side. Well, when there's a lot of cocoons into the walls of the cells, it's actually difficult for the bees to reshape that comb into a queen cell. Oftentimes that's when you'll see a queen cell that kind of really hangs off of the side, kind of out. They float the queen out and build the cell down because they can't reshape the cell walls. So what I did here is I looked for eggs and just that borderline of eggs and young larvae, and I made the cut here. So now all the bottoms of the cells are kind of torn open, and it's really easy for the bees to make queen cells along the bottom there, where they're actually going to be available for you to use without cutting up a whole bunch of comb. There are other ways to do this. Some people go in where the larvae are and they just kind of tear down the very bottom of the, with a toothpick or something. They just kind of dimple the bottoms of the cells. And that gives the bees kind of a head start in making the queen cells. So what I did here is I took, I think it was 2 deeps of bees. I trimmed the comb as I showed you in that last slide. And then I took, a little bit of food. I didn't have to put too much food. There was a flow on, and obviously I had foragers from 20 frames coming to that location, and I put them into 5 frames. You can see they can't even all fit in the hive there. You're going to really crowd the bees, because what causes the bees to want to swarm? They're overcrowded. They don't have any room to store anything. They don't have any room to do anything but to reproduce and to swarm. So you really crowd them. Now, you don't have to find the queen when you're doing this, because you're not trying to dequeen this hive. You're trying to tell this hive to swarm. Well, they do that with the queen still in the hive. So you don't even have, if you can't find the queen, you can do this and you can still raise queens. And then a few days later you can see that, again, the wire's a little bit confusing here, but right along the bottom there, I've got these queen cells where I can cut them out easily. In this case, they're a little bit close together. I probably could get 2 or 3 of those out without damaging the other, without damaging them. There are all different ways to do this, where you can squash, every, 2 larvae between every one so that they're spaced out properly. And it really depends on how many queens you want and how efficient you want to be. If all you want is to raise a queen, now you've got a frame with queen cells on it, and you're ready to go to make your split. Does that make sense? Okay. So I'm not going to go through all of these methods. This is kind of a history of queen rearing. And the Hopkins method, the Miller method, the Alley method, the Smith method, the Doolittle method. Frank Pellet wrote a great book on queen rearing where he did a very good summary of all of these. And when you read these methods, you see that it's actually an evolution of one step at a time to the point where we're grafting. The Hopkins method, you literally, if this is your comb, you literally suspend the comb a couple inches above the brood nest, horizontally. And then the bees draw the queen cells straight down from the comb. And it goes on until we're cutting strips of larvae out of the foundation and gluing them to the bottom of a piece of comb. And eventually we get to the Doolittle method, which is grafting. But all of these, and the most important reason to go through and to read all these methods and to understand them, and not just these other methods, but you go onto the internet and you read Michael Palmer talking about how he raises queens. You read Kirk Webster about how he raises queens. You read all these things, and what you start to see is that you're coming to the same point from all these different directions and all these different angles, because this is what it is that you want to understand, is that thing in the middle. Because once you understand that thing in the middle, you don't need a method. You can say, oh, I need some queens, and you look at what your resources are and where you're at, and you just make some decisions and you do it. But there are some common aspects to all of these. The bees must want queens. They're not going to raise a queen if they don't want a queen. So if you crowd the bees, well, they want to swarm, so they make some queen cells. If you take if you do a walk-away split, you take the queen out of part of that hive, and all of a sudden the bees don't have a queen, so they want to make one. You have to do something to make them want queens. They have to have larvae of the proper age. And these are generally larvae that are 18 hours or old or less seems to be ideal. I do know some people that graft from older larvae. Some of the literature says that anything less than 3 days old is okay. But I've been reading there's some recent study, I think Dave Tarpey, that looked at 18-year-old and younger larvae versus 2-day-old larvae. I'm not sure about all of his analysis, but it does seem clear that the queens were bigger when they were raised from the younger larvae. And I think there's a lot to be said for using the youngest larvae possible. And as Michael Bush says, you look for the little pool of royal jelly that you can't see anything in, and that's what you want to graft. There have to be enough resources available for the number of queens that you want to raise. If you want to raise 1 or 2 queens, you almost don't have to do anything. You're in Florida. You've got pollen coming in almost year-round. You've got some nectar coming in almost year-round. But if you want to raise 20 queens or 30 queens in a cell builder, you've got to make sure that they have food. And they have not only just food, but food coming in. And that's why I like to use that location and those foragers, because I really hate to feed bees if I can avoid it. But if I can give a whole bunch of foragers by using that location to a cell builder, then and there's something coming in, it isn't raining all day every day, then I really feel like they're being fed. And remember that there's honey that they've stored and there's nectar coming in. Well, that's like your paycheck versus the cans of food that you have in your pantry. The cans of food that you have in your pantry doesn't make you think that you can afford to go out to eat, or do luxurious things. But if you've got a big paycheck coming in, that's a different kind of resource that you have. And so you either want to be feeding the bees or you want to make sure that they're bringing in a good amount of food. And also the cells must be separable enough. Like that slide that I showed you before with those 4 cells in a row, those aren't quite separable enough to reliably get 4 queen cells out of that. Easily get 2 out of it by sacrificing the 2 in the middle. But you want to make sure that somehow they're separable enough in order to use them. So, and again, I have some of this stuff here that if when we're done, if you want to take a look at some of it. When I talk about grafting, Ramona, can you get my little bag of stuff? When I'm talking about grafting, what I'm talking about is literally lifting that very young larvae out of the cell that she's been laid in and putting her into an artificial cell that you orient vertically rather than horizontally to raise a queen. Here, I need the whole thing. So I'm just pass a couple of these around so you can see. These are artificial cell cups. These are the JZ BZ cell cups. And what I want you to notice is that they have a few different kinds, but I like these ones. If you put your finger on the pin in the back and you rotate the cup, you'll feel that it's not quite round. And that way you can actually fit it into a small kerf, like on a bottom bar. Or like, here, Sam brought this. These are some emerged queen cells. You can see that this is a bottom bar, and just the groove in the bottom bar, these cells go into. And this pin that isn't round means that if it's a little bit loose, you can just twist it and tighten it up in there. And it's quite nice.